लेके जावे का।
सर वो टर्मिनेट हो गया था, सॉरी सर। प्लीज ज्वाइन सर। हाँ। हेलो। अभी ज्वाइन कर पाएंगे सर? वो शायद वो टर्म। ओके। ओके ओके, थैंक यू। श्योर सर। सपा सकरम पाटिल, अनामिका सिंह, अंकित शर्मा, डॉक्टर नीलम मैडम।
Yes, sir.
यस मैडम।
Yes, sir, sir. I'm open to discussion if anybody wants to discuss about the biofilm. I'm particularly interested to talk to Dr. Arunima Biswas. Is she around?
शी इज हियर मैडम।
Yes.
Arunima, madam, I'm Neelam Kongwani. Basically, I'm assistant professor in marine biotechnology from Gujarat Biotechnology University. I'm also interested in work on the mangrove plants and their anti-biofilm activity. I have not worked with the honey, but I'm interested to work with the mangrove plants particularly. Do you have access, do you have you done any research on that part also?
No, I've it's only worked with honey from 5 or 6 different plant species of the mangrove
Okay.
area. Not on the plant extracts or anything, but yeah, definitely many groups are also working on that because it's like a treasure trove of research material. These mangrove forests are, yeah, really. So, ma'am, can I get your like contact mail ID or something so that we can maybe
Yeah,
collaborate in
sure.
the future? Yeah, okay, that will be great.
Yeah,
Thank you.
sure, because I found it interesting, how the honey, which is extracted, of the comb, which are present on the different plant, can have a different properties when we talk about their therapeutic or everything.
Yeah, so like honey, it's rich in sugar and peroxide, and also in polyphenol. So that gives them their antimicrobial as well as their antioxidant property. So I can send you like my published papers if you share your mail ID and also give you
I
some.
have shared my email ID into the chat box. I'm particularly interested, to work on the different area. So when it comes to the my research, I majorly work with the environmental relevance of biofilms, and I'm
Mhm.
also working on the bacterial biofilm. But I don't focus on only on the antibiofilm activity. I majorly focus on how the biofilm lifestyle can affect the antibiotic-induced persistence, which is one of the
Right.
most ignored part, because major of the studies only focus on the eradication of biofilm component, EPS targeting, and eradication of the biofilm. But what about the population which do survive under that particular stress and they can resuscitate even after the
Yeah,
certain period of time? So my research
exactly.
focuses majorly on the persister cells and how we can use the combination of natural compound in the with the antibiotics or maybe any bioactive compound to resuscitate the cells which are into the dormant state, so that we can protect the latent infection of any bacteria, like which happens in the case of TB. I majorly work with the Pseudomonas aeruginosa as a my model organism.
Okay. That's really very interesting because the persistent cells, I also feel like that is the crux of it all. How they, jump back when they get a favorable opportunity to grow back, and
Interestingly,
yeah, yeah.
we have not published a study. Like recently, few of my students started their dissertation work, and we came across this interesting finding that under the antibiotic stress within the biofilm or maybe in the planktonic cells,
Hmm.
there are cells which survive under the antibiotic stress. But after the removal of cells, there are only hardly few cells which can retrieve back into their original state. But when you add certain bioactive compounds, suddenly there is an increase in the rise of persister cells, which indicates that the when it comes to the combination of these chemicals, it's not only about the killing, but they also break the dormancy.
Okay, that's highly interesting. Okay, I get it. Okay. So basically forcing them out of the dormancy and then
Yeah,
targeting
it
them.
is one of the strategy which is one of the more ignored part when it comes to the biofilm.
Right, right, right. So, ma'am, yeah, I'll also share my mail ID and maybe we can discuss further later.
Yeah,
I'll
sure,
share
ma'am.
my mail ID on the chat box. Okay.
So have you targeted any column sensing related genes, madam? Dr. Neela, madam.
So, sir, recently, basically, I'm working in collaboration with the Indira Gandhi Center for Atomic Research, Kalpaka, and I'm also associated with NIT Rourkela. So we have, from the IGCAR Kalpakam, we have few quorum sensing mutant which I received from them. So the thing is, like this resuscitation, we basically did in the less quorum mutant. How
Mm-hmm.
the less quorum mutant in involved. So we basically, these are not the published data, these are the very preliminary data that we found. If the organism is having uh, uh, if we block quorum sensing, or if there is a quorum sensing mutant, it won't affect significantly the biofilm formation in that particular strain. But the thing is, after giving them the antibiotic strains, what cell do? They try to go into the dormant stage, right? And
Mm-hmm.
once you withdraw that antibiotic, they again start to regrow because there are hardly one or maybe less than 1% of the population which may survive under the antibiotic stress by going into the dormancy. Now, that dormancy period may last for a very long time. It may last for few hours to few months or maybe few years. The thing is, particularly the organism which is having the quorum sensing mutant, usually they lack this signaling to decide what is the exit point when to come out of this dormancy. So this is might be the one of the strategy. Rather than targeting the quenching part of the quorum sensing, we can target the resuscitation part so that our antibiotic current stock of antibiotic can work more efficiently.
Purnima, madam has shared her.
Yeah, thank you, ma'am.
It would be my pleasure to have some discussions with you on this
So,
topic.
madam, here
And Biprang sir, if you can join us also, maybe yeah, because you are also an expert in this field, so it will be great if we
so
can
it's
get
my
together
request to
sometime.
Dr.
And
Neela, madam, because here several students are here, madam. So if for the students, if you can explain that how to means go for the essay of the biofilm inhibition or essay for the quorum sensing phenomenon.
Okay, so
Why?
basically
Because
when we
because the students are here, madam.
okay, so when we look into, particularly when we look into the biofilm, they are around. Even your dental plaque to the slime which you look into the around, they are everywhere. And the most of the organism when they come in contact with any surface where they can attach, they start to form the biofilm. Now, when we look into the practicals or the common protocols which we usually do for the biofilms are the basic glass tube assay, which involves the growing cells under static conditions and then staining that glass tube with the crystal violet. This is one of the most basic screening we do for the biofilm screening. And then de-staining it with either methanol or maybe with 33% acetic acid or ethanol. Now, once you destain it, there is a portion of dye which remain bound to the EPS matrix of the biofilm. If you can see there is certain ring or something around the biofilm, around the tube, it indicates the attachment of cells. This crystal wallet technique is also reproducible, or you can also use the microtiter plate, which provide the micro environment for the biofilm growth. When you go to the more advanced version, it is recommended that when we are particularly studying the biofilm, there are the multiple microscopic techniques which actually help us to quantify the biofilm. So the first basic technique is the fluorescent microscopy. Now, the fluorescent microscopy, we use a combination of different fluorophore and the fluorescent stains, which give us idea about the attachment. Based on the coverage of a biofilm, we can determine what is the thickness of biofilm, and it give us the uh, information related to the biofilm growth in terms of raw integrated density. Moving beyond fluorescent microscopy, there is a confocal microscopy. In the confocal microscopy, it is also based on the staining of the biofilm. But the thing is, using the confocal microscopy, we can have the idea about the three-dimensional structure of the biofilm. And particularly, we get the Z-stack with the confocal one, and we can have the exact idea about the what is the biofilm thickness. Now, the whatever the data set we get from the COMSTAT are then processed with the multiple tools, for example, Biofilm Q or COMSTAT, which then give us the complete biophysical information related to the biofilm. Now, these tools are designed with certain algorithms, and even you can calculate what is the roughness of biofilm, what is the colony microcolony size of the biofilm, what is the diffusion distance, what is the minimum and maximum microcolony length, what is the total biomass within the biofilm. So all these parameters can be easily get if you have done certain confocal studies and process those images with the biofilm tube or COMSAT. Apart from that, AFM will also provide information related to the thickness of the biofilm. And simultaneously, number of people also study the biofilm using the scanning electron microscopy. So particularly in the scanning electron microscopy, we have more idea about what is the attachment between the cells and what is the exact mucoid structure of the biofilm. Interestingly, I have one publication from 2013 in the Panibacillus luteus biofilm, which was isolated from the mangrove sediments. And we came across that within the biofilm, if you use a high-end biofilm, you can even able to trace the phenotypic switching of the cell within the high population density. So this high-end microscopy even give us idea about what is the phenotypic diversity when it comes to the biofilm. It's not only about the EPH, it's not only about the protein component or maybe the extracellular component and the cell, but there are also the phenotypic diversity within the biofilm. So when we study the biofilm, it is always recommended to use the multiple techniques to come across certain conclusions.
Thank you so much, Dr. Neelam Madam, for your sharing your insightful research experience, and it was truly the inspiring and enriched our understanding of the subject that is the biocell. So, students or the participants who have any query to Dr. Nilma Madam, you may ask now. If you have any query regarding the biofilm,
Thank
Message
you.
or Pankaj Bharti. Hello.
Yes, sir.
Yes, Pankaj Bharti, you are audible.
सॉरी सर, एक्चुअली मैं पहली बार जुड़ा हूँ तो इसका माइक
हम्म,
ऑफ करने का मैं देख रहा था।
अच्छा अच्छा। So is there any question or query for the Dr. Nilima Madam? Dr. Neela Madam. Oh, thank you, madam. So Dr. Rupa Niroli is here. Dr. Prasun Kumar.
Good evening, sir. This is Dr. Rupa Niroli talking.
Good evening, madam.
So, I have worked on antimicrobial resistance. So, I was interested and keen on this topic, how biofilm will contribute to the resistance. So, I just joined this session for this.
Mhm. Now I request you to share your research experience on the antibiotic resistance.
Yes, sir. Yes.
You may share your research experience here, madam, on the antibiotic resistance.
Uh, like last time I shared in detail, but I guess those participants were different. So, if you feel I should share, then I'm happy to share my research, sir.
Yes, madam.
Huh. So basically, I have worked on isolation of antibiotic resistant organisms like E. coli I have worked on, and I processed the fish samples and fish waste as well. From those, I isolated E. coli samples, and I was keen on beta lactamases, and I found three genes. I mean, my area of interest was my focus was on three genes, that is CTX, that is beta lactam producing CTX, SHV, and TEM. So I performed PCR studies, and again ERIC PCR also I performed. I mean the results were little astonishing. Like CTX is so very much prevalent in most of the studies, but in my studies the um, like SHV and TM were also most prevalent than compared to the other studies. So this was my topic of interest, and I did my PhD on this. If any queries regarding this can be answered, sir. Thank you.
So have you ever tried, madam, with synergistic effect of the any natural compounds with antibiotics, just to minimize the
Sir, basically,
of
uh-huh.
antibiotics?
Like in our lab, like we have worked on the remedy also. We
Mhm.
found the solution also. We have isolated bacteriocins, that is microbial peptides, wherein my organisms were showing resistance to mostly 13 antibiotics out of 14. Only it was sensitive to cholesterol. So such organisms I was having. So we checked with the bacteriocins, and this bacteriocin was able to inhibit the bacterial growth. That is, that those bacteria are sensitive to our bacteriocin. So it was our output for the study.
Okay, thank you, Dr. Pawan. Dr. Pasun, sir,
Thank you, sir.
Pasun Kumar. Kumar Pallavi, Pooja Sandesh.
Hello, sir. Good
Hello,
evening,
sir.
everyone.
Hello, good evening.
I'm Sandesh. I'm currently pursuing my PhD from Manipal University, Jaipur. So I joined this session because I work on bacterial biofilms for my PhD. Basically, my area of like my PhD research involves around bacterial two-component system, which is involved in like production of various virulence factors such as like I work on MRSA, so it produces multiple virulence factors, and it is a strong biofilm former. I am in my preliminary stage, like I just now I have joined PhD. I have been doing biofilm assays, like 96 well plate assays. I have got one or two queries, that's why I have joined this session. Sir, may I ask the question?
Sure, sure.
Thank you, sir. So, when I was performing biofilm assay, like when we perform in a test tube, we take round bottom test tube. So, what I was assuming is that if I use a round bottom 96 well plate, the results will be same. But what I have noticed is that my cells are not evenly growing in case of when I am using round bottom plate. And like I have performed, I tried doing it multiple times, but I have.
Sandesh,
Huh.
yeah, I have answer to your question. The reason being, usually in most of the aerobic microbes, or particularly the bacteria, what happens, the maximum biofilm formation usually is visible at the liquid air interface, right? So that's why when we test them, we usually recommend to see if there is any presence of ring. When you even you perform the microtiter assay or in the glass tube assay, visual observation are usually done on the presence of ring. The second point is when you look into the glass surfaces and the microtiter surfaces, both are different substratum. It is made of silica and it is polystyrene. Now, the attachment property is largely dependent on the property of the biofilm. That's why when we study biofilm, we always say when it comes to the biofilm formation, there are three major factors. They are physical properties of the surface, chemical property of the surface, and third one is the biological property of the organisms. So when the reason being when you are replicating two results in the different surfaces, you will get the different results. Another thing is when you look into the 96 well plate, majority of what happens, there are certain calcium treated and non-calcium treated. Usually, the calcium treated are available when you use these plates for the culturing of the animal cells. So, it has been observed that if you use the calcium treated 96 well plate, the chances of getting bacterial attachment is more as compared to the non-treated plates. So, largely, if you want to screen the biofilm, you need to take consideration on the surface property or the surface you are growing. The second solution to this is once you are if you want to quantify, if your focus is on the quantification of the biofilm, although the literature suggests that you can grow on the microtiter plate or the glass tube assay, the alternate way to quantify in the term of destaining is grow them in the glass tube, destain them with the appropriate destaining agent, then quantify on the basis of absorbance of that destain solution that you can do on the microtiter. So if you are planning for the screening, cellular attachment, it is largely depend on the substratum on which the biofilm formation is taking place. Hopefully that answer your question or maybe the problem.
Yes, ma'am. Thank you for this detailed answer. Ma'am, like I have another thing is to ask, that is I have several inhibitors. Like we have designed inhibitors competition design, in the sense we have screened from existing library, and we have synthesized also few of them based on their parent chemical structure. So when I'm screening them, like I asked many of my colleagues, like I'm the only one who is starting that biofilm related things in my lab. But I
Mm.
asked my previous classmates and my seniors. So some of them are suggesting that you use treated flat bottom plate, and some of them are using don't use treated plates. If I use treated plates, they will promote the adherence of bacteria to the plate.
Yes,
But I
yeah,
want to
that's what.
naturally see, but
Mhm.
I'm sorry. I want to naturally see whether the bacteria is attaching to the surface or not, and all that.
So the same thing I told you, na, it largely depends on the property of the surface. You look into the any surface. For example, you submerge iron coupon into your water or maybe the culture, or you submerge some steel coupons or maybe the metal. The attachment would be entirely different, right? That's why people make the anti-biofilm or anti-adhesive substance. So the whatever, like when you are growing or screening simply on the basis of the microtiter plate assay, it will give you false positive result. That's why it is always recommended. The standard surface for testing the biofilm growth is basically the glass. But if you are not doing any study related to the surfaces, you can choose either calcium treated one or untreated one, it won't affect. But if you are using the untreated one, the problem is the coating material they have, it is somehow made up of the anti adherence chemical that won't allow cell to attach. However, if you are using the treated one, it promote the attachment of the cell. Now, the second thing, what you told that you are looking for the inhibitors, biofilm inhibitor. That's why it is always recommended if you are using for, if you are focusing on the biofilm inhibitors, you need to also focus on a microscopy based technique of the biofilm, because the reason being this crystal wallet assay or attachment assay is simply based on the visual observation and the semi-quantitative analysis based on the staining crystal wallet, which may give you the false positive result. But once you observe your biofilm under the microscopy, it will give you the complete information related to the surface topology. Like if you stain them, you can use a different combination of stain to see which component is present at which particular time point, what is the live dead cells, what is the EPS composition or anything. Any tau, whether you're doing anti-biofilm study, after doing the glass tube work, microtiter plate work, you need to prove it with the microscopic based observation. Otherwise, it won't be a good observation, or you won't have a concrete idea about what exactly is happening when it comes to the anti-biofilm or anti-attachment property of your compound.
Okay. Thank you, ma'am. Thank you. That was again another like very like good answer to my question, ma'am. I am also planning to perform confocal of the same experimental setup in which I'll check the antibiofilm activity. Um, so, ma'am, I'll try to incorporate your suggestion. But ma'am, another thing is that I have multiple inhibitors, so I have to screen them. I have to screen a lot of
Yes.
inhibitors. So I'm just wondering that we just like our paper mostly focus on the computational aspect, and we just have to show
Mhm.
the whether it is inhibiting biofilm or not, and including confocal sophisticated microscopy observation along with that. That is, ma'am, we don't have that facility. That's why I'm bringing this point. That
Yeah, you can even, and that's what I'm telling you. What usually, like, initially when I started working with the biofilm, I realized, see, my work was majorly focused on, complete PhD was focused on the biofilm, role of colon sensing in biofilm formation and its interaction with the pollutant. And even till today, I'm working with that biofilm inspired biomaterials. Now, the thing is, on the surface on which you are studying the biofilm is extremely important. When my application demands me to use a biofilm for the bioremediation purpose, the thing is, my major focus is to increase the biofilm growth. So when I look into the polystyrene surface or the glass surface have the more adherence property. That's why the population will be more, biofilm formation will be more. So I particularly choose that subjects. But now when you are looking into the antibiofilm, you need to focus on few things. What is your target antibiofilm area, right? Surface. Now, most of the people go with the 96 well plate. The reason being, you grow the biofilm, you will have a visual staining, and same plate you can use for the quantification after destaining. But it gives false positive result many time. Alternatively, what you can do, you can grow the biofilm on the glass tubes, you can destain it, and that destained solution you can quantify in the microtiter plate. But yeah, you are commenting, even if you look into the manuscript these days, targeting any antibiofilm study simply on the basis of microtiter and the glass tube won't be feasible. Even you can look for another study which might not give you the exact quantification, but it will give you about the composition of biofilm. For example, if you are growing your biofilm on the suppose on the glass surface and you want to see whether your compound is effectively working or not, you can do two more things. One thing is you just go for the time killing assay. It means like at a particular time point, maybe 0, 6, 12, or maybe 24, 48 hours, whatever your time points are, you can go for the viable cell count. The second thing is at all different time point, you can just extract the extracellular polymeric substances which are present at that particular time point. You can go for simply the this one, the carbohydrate, total carbohydrate and total protein quantification. It will give you idea how your compound is acting. So even if you don't have the high-end instruments, you can combine all these biochemical processes and biochemical studies, and you can then predict that that it's not only about the quantification, visual quantification. It is also affecting the live cell population. It is also affecting the overall biomolecular component of the biofilm. As we all know that the biofilm is nothing without the extracellular matrix, right? So these are the additional things which you can incorporate if you don't have access to any high-end microscopy techniques.
okay, ma'am. Ma'am, thank you so much for your insights. Ma'am, I'll definitely refer your PhD articles. Like you mentioned, you worked on quorum sensing. My work is also related to like that only. I will definitely refer your paper. Thank you, ma'am.
Welcome, Sandesh.
Madam, I have one question, Madam. Dr. Nila Madam.
Yes, sir. Yes,
Means,
sir.
in addition to the confocal microscopy,
Hmm.
means how can you go for the means imaging of biofilm?
Okay. So, sir, there is again the same thing. If you don't have access to the confocal microscopy, the
Mm-hmm.
fundamental things usually most of the people do is they, excuse me, they go
Mm-hmm.
with the fluorescent microscopy. Now, the difference between the fluorescent and the confocal is although they both have you use the same fluorophore for the both the cases, but thing is in the fluorescence you will have the total fluorescence of the biofilm, and using the combination of different, if you want to target suppose carbohydrate, protein, nucleic acid, live or dead cells. Based on the fluorescence intensity, you can have the information related to the arbitrary information related to the fluorescence, and you can channelize whether which component is present. So after these two methods, glass tube assay and the biochemical composition quantification, the third phase is always fluorescence microscopy. After that, there are other things which come into the picture. And recently, many people also integrate biofilm study with the flow cytometry study just to target metabolically active cells, which may include persistent cells also, but they may be into the dormant cells. So there are certain high-end instruments which give you idea about what is the population of live cell, dead cells, and the cells which are viable and unculturable, which is the third dimension of biofilm. Even any cell which is present during the stationary phase, which indicates the dormancy.
So, thank you, madam.
Excuse me, may I say a
Sure,
few
madam.
words, please? Yeah. So, Neelam ma'am, thank you so much for all your inputs here. And with Prang sir, I was wondering, like, if we could arrange maybe a national level seminar on biofilms and basically biofilm research, because and we can have invite Neelam ma'am as an expert there, because these troubleshooting things, they are very important for the PhD scholars and also for researchers like us. And she's such a great expert. So maybe we can actually arrange a national level seminar on biofilms, because this topic, like, it is a really a hot topic in today's research among microbiologists. So maybe we can arrange that sometime.
Sure, madam. We will try for it.
Neelam ma'am, we are already booking you as a speaker.
Okay,
Because the
thank.
most problem that Dr. Neelam madam has explained that in most of the biofilm research, what are presented in the research articles are the false reading. Only by the title plate, most of them in the can say the state level what the research are going on. Most of them are presenting only by that title plate essay. So that may be false reading.
Hmm.
So actually, the thing is, you learn
Mhm.
once you face the problem. So the thing is,
Sure.
what I realize, I am somebody who is working on the both the dimension of the biofilm, positive and the negative. So the
Mhm.
thing is, when I started working on the biofilm, I focused more on the positive aspect. So my focus was how to grow them, like how to grow them in a very good manner. And when we started screening the surface, we realized that okay, this is a problem. And when in 2010, when I initiated this work, even I was not aware about many things. And somehow I got a good mentor from the Indira Gandhi Center for Atomic Research, even the student who are available here. And if
Mhm.
you are into the your MSc, maybe the second semester or third semester, you are looking for research in the microbiology, I recommend if you are like interested in the biofilm, you can approach this biofouling and biofilm processing section at the Indira Gandhi Center for Atomic Research, Kalpakam. They have a complete facility related to the biofilm, how you can study the biofilm related to the biofouling, marine fouling, for the bioremediation, and even under the clinical conditions. So, like you can look for the biofouling and biofilm process section. So that is the section which basically work on all these dimensions. So somehow I was also exposed to these mentors who taught me everything. But when we look into the clinical part of the biofilm, we always focus on how to restrict their growth. We never focus on, for example, you take the contact lens surfaces, they are made up of the different hydrogel. Once you screen them, you will all have a different attachment properties of the biofilm. And this is the reason when we talk about the antimicrobial treatment, we only focus on the planktonic cell, but we always overlook on what surface actually we are targeting. So this is something who like it maybe comes with the learning, but yeah, these are the something which major of the people who are working on the antibiofilm ignore the surface properties.
Hello.
Yes, sir.
Yes. So thank
Ma'am,
you
I
so
have a
much.
one query.
Yes,
Hello.
Pankaj Bhatku.
जी सर,
Mhm.
सर आई एम डूइंग पीएचडी इन बायोटेक्नोलॉजी एंड माय रिसर्च टॉपिक इज बायोफिल्म। मेरा एक क्यूरी था सर कि मेरा बायोफिल्म में मॉलिक्यूलर डॉकिंग कंप्लीट हो चुका है। अब मैं मॉलिक्यूलर को सिमुलेशन के लिए दिया हूँ, तो क्या मॉलिक्यूलर सिमुलेशन तक मैं एक रिसर्च पेपर पब्लिश कर सकता हूँ?
मीन्स बायोफिल्म के लिए मॉलिक्यूलर डॉकिंग किए हैं।
हाँ, प्लांट नेचुरली प्लांट कंपाउंड को आइडेंटिफाई करके मैंने डॉकिंग तक का प्रोसेस वर्क कंप्लीट कर लिया है। अब
आपका
मॉलिक
प्रोटीन टारगेट कौन सा था?
मेरा बायोफिल्म का जो एग्लूटिनिन लाइक एल
हम्म
एस 3 प्रोटीन है,
हम्म
एग्लूटिनिन लाइक प्रोटीन जो है, एंड सैप 2 प्रोटीन, ये सब टारगेटिंग प्रोटीन है सर बायोफिल्म का। डॉकिंग वर्क हो चुका है, सिमुलेशन के लिए मैंने दिया हुआ है, तो सिमुलेशन तक का एक रिसर्च पेपर पब्लिश कर सकता हूँ क्या?
मैं समझता हूँ कि डॉक्टर नीलम मैडम कैन सजेस्ट बेटर।
जी जी मैम, ठीक है।
एक्चुअली पंकज वी फेस द प्रॉब्लम, तुम्हारे जैसा ही हमें भी प्रॉब्लम हुआ था। वी हैव सर्टेन स्टडीज ऑन द मैंग्रोव फाइटोकेमिकल्स। हमने स्पेसिफिकली बायोफिल्म रिलेटेड ईपीएस जो जीन होते हैं उनको टारगेट करके किया था, बट दीज डे जनरल्स आर नॉट एक्सेप्टिंग एनी कंप्यूटेशनल स्टडीज बायोफिल्म में, तो खास करके अनटिल एंड अनलेस आपने उसको इन विट्रो प्रूव किया है कि नहीं। फिर वो इन विट्रो आपका अगर माइक्रोटाइटर प्लेट ऐसे या ग्लैड ट्यूब ऐसे है वो भी चलेगा, और मे बी द टाइम किलिंग कर्व भी इंक्लूड कर सकते हो। द सेकेंड थिंग इज कि अगर आपके पास में ये नहीं किया है आपने, तो सेकेंड थिंग जो है, द फाइटोकेमिकल ऑन विच यू आर वर्किंग, इफ यू हैव एक्सेप्टेड देम और आपके पास जीसीएमएस और एलसीएमएस का कोई डेटा है, तो
जी
आप उसको कोरिलेट करके यू माइट गिव अ ट्राई। बट जो ये जो है एंटीबायोफिल्म के लिए ग्लास ट्यूब ऐसे टाइम किलिंग
जी जी।
ऐसे है, ये तो कोई भी कर सकता है। दीज आर द सिंपल माइक्रोबायोलॉजी बेस्ड टेक्निक्स, तो ये एक्सपेरिमेंट यू माइट नीड टू इंक्लूड, अदरवाइज इट वुड बी डिफिकल्ट।
ओके मैम, थैंक
मीन्स
यू मैम।
अच्छे जर्नल में पब्लिश करने के लिए मैडम जो सजेस्ट किए वो करना होगा,
यस मैम,
क्योंकि
मतलब
सिर्फ मॉलिक्यूलर
सिमुलेशन के बाद
डॉकिंग।
मुझे इन विट्रो एक्सपेरिमेंट परफॉर्म करना
श्योर
है।
श्योर।
हाँ।
ओके, थैंक यू।
तो ना Kumar Pallavi, Pankaj Sandesh. So we are very fortunate that today with us Dr. Neela Madam is here. So if you have any query, may ask to Dr. Neela Madam, or Dr. Arunama Madam is here. So I think there is no any more query. So thank you all the participants, Dr. Neela Madam, Dr. Arunama Madam, for sharing your insightful research experience. So truly it was the inspiring one, and I think that most of the participants that have joined today learned a lot about the biofilm. And my heartfelt thanks to all the participants for joining and contributing to this engaging session. So looking forward to our next session together. So thank you once again. Thanks to all of you for joining today's session.
थैंक यू सर।
थैंक यू
Thank
डॉक्टर
you
नीलम मैडम।
all.
मैं आपको एक मेल करूंगा क्योंकि मुझे एक प्रॉब्लम है बायोफिल्म वाले ऐसे में। हेलो।
जी सर। जी सर। जी सर।
Hello, ma'am. Can you please share your mail ID? If we have any query, we can ask you or
Yeah, my email ID is already in the chat box probably, neelam.kungwani@gbu.edu.in.
Just I am pasting here again. Sandesh, just I copy paste here, okay, in chat box.
okay.
So thank you once again for joining today's session, and we hope that we get Dr. Neela Madam, Arunima Madam, and all participants in our next session also.
Sure, sir. Thank you.
Yes,
And
sir. Thank you, sir.
Viprang sir from West Bengal MBSI unit, we will arrange for a national level seminar on biofilms. Okay, and
Yeah,
we will
at least
invite Dr.
we can
Neelam
try
to be
for
a research
that
professor.
online mode.
Yeah, online. Sure, online
Yeah.
mode. Yeah. Yeah. Okay. Then participants from different parts of the country can join. No, so that will be great. Okay.
Our
Thank you.
keynote
Thank you all.
speaker is fixed now.
Yeah.
So thank you so much, ma'am.
Thank you and good night to you all. Bye bye.